I'm Lisha Shao. I started my lab in the Department of Biological Sciences last January. Before joining UD, I was a postdoc researcher at Geneva Research Campus of Howard Hughes Medical Institute. My lab studies the mechanisms of reward using fruit fly Drosophila melanogaster as a model system. So today I will talk to you about our motivation and the research plans for the INBRI pilot project that pursues the interaction between social stress and reward. Okay. So before delving into the interaction between stress and reward, I would like to talk about why we are interested in how the brain perceives and processes rewards. To survive and reproduce, it's important for an organism to seek for food, water, sex, and social interaction. This is true to all the animals, from humans to flies. The brain has evolved to ensure these meaningful stimuli are perceived as pleasurable or rewarding, so that one will be motivated to obtain more rewards and maximize their chance to survive and reproduce. However, at pathological conditions such as addiction and depression, the same brain structure that takes us to pleasure can also put us in agony. My lab will take advantage of the tractable fly model to pursue the following questions in reward mechanisms. First, we would like to crack the neural pathways that mediate different meaningful stimuli, such as food and sex. Next, we want to understand how different reward-driven behaviors interact by characterizing the architecture of these pathways. We also want to determine how these pathways are modulated by internal states and environmental stresses. The INBRI pilot project will specifically address how brain reward system is modulated by social stress. There are various types of social stress, such as being beaten up by another conspecific or being rejected by a mate. We are interested in a more common and basic stressor, extreme social density, such as social isolation and crowding. As social creatures, we are particularly vulnerable to extreme social densities, such as loneliness and crowding. The exposure to these psychosocial stressors causes both physical and mental health issues. For example, according to a 2019 national survey, 61% of Americans are classified as lonely. It's been shown that loneliness is associated with a 26% increase in the risk of premature mortality. And this epidemic of loneliness is exacerbated by the social distancing and quarantine in the ongoing pandemic. On the other hand, although less studied, overcrowding has become a health threat in developing countries and special living conditions such as refugee camps. The negative consequences of extreme social densities have an evolutionary basis. The primary human ecology is a social ecology. Being in an optimal social network is our baseline state. Deviation from this baseline compromises our survival and reproductive fitness. This is true to animals, even the ones that are not considered as social animals. Social isolation leads to lack of conspecific support and mating opportunities. It also increases individual vulnerability to predators. On the other hand, crowding causes increased competition for resources and mating opportunities, and normally results in increased aggression. Crowding also elevates risk of disease transmission. So we can learn the biology and gain insights into how social density is processed and how social density modulates behaviors from animal studies. Here are the questions that we aim to address in the pilot project. First, what are the behavior consequences of extreme social density on motivated behaviors? So we will focus on mating and feeding. Second, we aim to find out how the intensity and dynamic of social densities are detected in the nervous system. For example, what sensory modalities are critical for the detecting of social density and how the duration of exposure is encoded. Also, we want to know how does the deviation from optimal social density regulate brain reward systems through the activation of stress responding system. So previous studies in flies revealed the key genetic and neural components in stress responding and reward pathways. We will focus on the interaction between them. The stress and reward system is, so in mammalian, the stress and reward pathways in mammalian nervous system are highly complex and involve millions of neurons. It's fairly difficult to figure out how they work together with the desired details. While it's challenging in the mammalian system, we can step back and take a look at what a simple brain can teach us. The fruit fly Drosophila melanogaster has a brain with only 100,000 neurons, of which about 200 neurons are dopaminergic neurons that are critical for reward function. For your reference, here is a comparison of sizes of a human, a mouse, and a fly brain. Although considered as very simple creatures, flies have enriched behaviors. So they eat, drink, have sex, like alcohol, and get addicted to it. Besides, fly model has very unique and powerful genetic tools that allow observing, activating, silencing neural activities, and manipulating gene expressions at single cell type resolution. There are also advantages that are specific to the study of social isolation and crowding. Flies' short lifespan is suitable for study the effect of chronic stress. It's also very easy to titrate the social density of flies and the duration of exposure. So it's economic and suitable for high throughput experiments. So probing the link between stress and reward pathways in flies becomes very attractive to us. So this is the research aims for the pilot project. First, we will determine the effect of social stress on reward-driven behaviors and from different perspectives. So we will establish the research pipeline, examine the sexual dimorphism and temporal and quantitative effects of social stress, and determine the sensory modalities that mediate the effect. In aim 2, we will probe the link between reward and stress systems. So we hypothesize that neuromodulators may play a role in bridging these two systems. This will be tested by specific neural and genetic manipulation. We will also identify more candidate genes by ongoing RNA sequencing experiment. Specifically, we expose flies to different social density, social environment for various duration before testing them in the different behavior tasks and assays. The mechanisms will be determined by examining these tasks and assays after specific neural activation, blocking, or gene manipulation. The project is still at an early stage. I will show you some preliminary data of how social stress affects fly reward-driven behaviors. In the first experiment, we tested flies' motivation to mate to test if their motivation to mate is affected by the exposure to social stress. So here we pair males after different social treatment with virgins and found that males showed much stronger motivation to mate with females after social isolation and crowding. In the second experiment, we tested the effect of social stress on flies' ability to take risk. Here we let flies cross gaps with increasing width that becomes more and more difficult to cross along the distance. We found that after social stress, the flies were more reluctant to cross the wider gaps, suggesting that they showed less motivation to take risks. Last experiment is to test the effect of social stress on flies' preference for artificial reward and punishment. So here we express a red-shifted channel rhodopsin in neurons whose activation is known to be rewarding or punishing. When flies were group housed, they show preference for the activation of the reward neurons and show avoidance to the activation of the neurons of punishment, such as beta receptor neurons. However, after the social stress, they show stronger preference and stronger avoidance to the activation of the same groups of neurons. So these data basically suggest that chronic social stress significantly regulate flies' reward-driven behaviors. So next, we will dive into the mechanisms and figure out the genetic and the neural pathways that mediate this regulation. So I hope to update with you the new discoveries next time. So it takes a village to start a lab, and it takes two during a pandemic. So I have many to thank. First of all, I want to thank all my lab members for their work, and I want to thank the colleagues in my department for their mentoring and the support. I also received a lot of help from folks at Genelia. Finally, I want to thank the support and opportunities from all these funding agencies. Thank you for your attention, and I'm happy to take questions.